So for this session, participants will learn how to identify dengue vectors, and we will be sharing some challenges that we encounter when we use different traps for identification. And also, we will be sharing how we can share this specimen or store them or transport whenever we need certain help for validation or that we need it for certain additional experiments. So I understand there are many entomologists attending this session, and maybe later you can share your own experiences in handling your mosquito samples. So mosquitoes can be identified in different life stages. However, there are certain stages that are more accurate for use in identification. These will be the adults and the larvae. However, for eggs and pupa, you can still use them for identification, at least to the genera level. So for adult mosquito identification, how do we know if what we're handling is actually a mosquito? You can write your answers in the chat if you have some idea. So what makes a mosquito a mosquito? This is for adults. Any suggestions? So for mosquitoes, they are really quite distinct actually. Uh, the first thing that you have to look out for will be the presence of a long proboscis. So you can see from the picture there is a red arrow. Above it will be the two maxillary pulpus or pulpi. This is actually a photo of Anopheles mosquito. So we just wanted to show you that sometimes the pulpi is long enough, as long as the proboscis, so you may mistake it as the proboscis. Normally, if you're dealing with culicin or mosquitoes that belong to genera like Aedes and Culex, normally the pulpi will be much, much shorter than this. Any other suggestion? There will be a few characteristics that we look out for when we consider an insect as a mosquito. Another will be the body and the legs would be covered in scales. Now, this would later I will share with you that we are currently actually trying to differentiate mosquitoes from other insects. So these are the characteristics that will differentiate them. So most insects, it should be a combination of these characteristics. It cannot be that you see an insect that it has a proboscis, it will already be a mosquito. No, it has to be combined with other characteristics such as these, which is the body and legs are covered in scales along with a long proboscis. Yes, as an insect, it will have three body parts. So you have the thorax, head, thorax, and abdomen. That is true. Agree. But also, when you look at the wings, these will also be covered in scales. So the wings are long but narrow. So, and then you will see that these are covered with scales. Other insects will have hairs covering the surface of the wings, or actually it's just a membrane, or sometimes the wing is of a different type of wing, which is what we call the elytra, that is for the petals. So, what is the difference between these scales and hairs? So that we can determine that the sample we're handling is really actually a mosquito. So these are examples of hairs and scales. What is the difference that you see in them? You can type your answers in the chat box. It's really hard to tell because when you see the scales, there are so many different forms of the scales. There are narrow scales, serrated scales, rounded, forked, broad asymmetric, many types of scales. But you notice that the hairs are just elongate structures. So, what is the first thing that we notice here is that when you look at the base of the hairs, it is usually wider than the tip. So this is actually because the hairs always taper to a point from the base. Whereas if you look at your scales, the scales will be narrow at the base but wide at the tip. So whatever form the scale is, it will always be more narrow at the base or where it is connected to the body and wider at the tip. Yes, we have differentiated the mosquitoes morphologically. And also, that is true. Wing venation is also characteristic of mosquitoes. It's just that um, when you want to zoom into species, it is more important to know the venation. But if you just want to know whether this is a mosquito or not, then the top three characteristics I've mentioned, which is the proboscis, the scales on the body and the legs, and on the wings, is the most important characteristics to determine them as mosquitoes. So, how do we recognize or differentiate Aedes mosquitoes from other mosquitoes? Any suggestions? Many mosquitoes do have spots on the legs, but not all of them. And also, some don't have any ornamentation on the abdomen, but some do have bands and other hair tufts or clusters of hairs in different parts of the abdomen. Body color and pattern is actually more important when you're zooming into species. So how do we recognize Aedes mosquito? The first thing to look at is the thorax. So you look at the back of the thorax and you look for the trilobe scutellum. So where is the trilobe scutellum? So you are looking now at the scutum. So this is the back of the mosquito. And then from the scutum, you go down further down and you will see the scutellum. How do we know this is a trilobe scutellum? You see the shape, it has like three scallops. Normally, many mosquitoes are like this. So this is a shared characteristic. So you have to look for additional characters to tell you that you are looking at an Aedes mosquito. Aedes mosquito is not limited to Aedes aegypti and Aedes albopictus. There are many Aedes species all over the world. So now, in addition to the trilobe scutellum, you also have to check for the presence of postspiracular bristles. So when you look at the side of the thorax, right, it's hard to remember where is the postspiracular bristles. So the first thing you look out for is the spiracle. So this spiracle is actually a hole on the side of the mosquito. This is how they respire or breathe in air. And then behind it is a teardrop shaped structure. Now, from this teardrop shaped structure, this is what we call the postspiracular area. The postspiracular area actually have hairs coming from it. So those three red lines is actually showing you the bristles on the postspiracular area. The white scales below it is not considered because they are scales and not hairs. So bristles are supposed to be hairs. And finally, the last consideration will be to look at the base of the hind coxa. The hind coxa is the attachment of the last leg to the thorax. So when you look at the hind coxa, let me see this. Okay, when you look at the hind coxa, this is, oops, sorry. This is the hind coxa. The base is I marked the base with a red line above it. And then it should be just behind the mesomeron. So I'm tracing it with a yellow triangle. That is the mesomeron, and the base, I'm marking it with a red line. So you can see that the base of the mesomeron and the base of the hind coxa are not in one line. They are two parallel lines. So this is what we call the bases are not aligned, and this tells us that we are dealing with an Aedes mosquito. It has to be a combination of all these three characters and not just one. And we cannot just choose one of them to say that this is an Aedes mosquito. So after you see that, okay, I'm dealing with an Aedes mosquito, you have to remember that there are many kinds of mosquitoes. So just the ones that you normally find around households is already 3 kinds. So you have Aedes aegypti, Aedes albopictus, and Culex kinkyfasciatus. I see from the chat that yes, the thorax will have a silvery line that will usually correspond for Aedes albopictus, but we do not limit ourselves to that just one characteristic. So to differentiate further between Aedes aegypti and Aedes albopictus, so these are the primary characteristics you look out for. The first thing is to look for the lyre-shaped marking on the scutum of Aedes aegypti and the single white line on Aedes albopictus. However, if you remember from Dr. Chong's presentation, with the Gravitrap, there is a sticky lining. So when you have a sticky lining, many of these characters or these scales will turn black when they come in contact with the glue, or they just simply drop off. So there must be other characteristics you can look for when you want to differentiate between the two. So when you're dealing with Aedes aegypti and Aedes albopictus, first thing you look at is the head. Yes, when you see the maxillary pulpi, they both have white scales at the tip. But you look at the base, towards the base of the maxillary pulpi, then you will find the clypeus. On the clypeus of the Aedes aegypti, what do you see from this picture? Yes? Do you see? Come again?
Please write in the chat box if they can write
Yes, please.
the answers would be better.
Thank you. There will be white scales on the clypeus of the Aedes aegypti, but for Aedes albopictus, there are no scales at all. It's actually what we call bulb. However, I must caution you, you have to check your mosquito first if it's male or female, because I noticed that the males, if you want to check, just try it yourself. Males of Aedes aegypti don't have the white scales on the clypeus. So you may be confused and think that this is Aedes albopictus, especially if the lyre-shaped marking is already scrubbed off or dropped off. So you make a mistake. Make sure first that your mosquito is a female. So this is only true for females. In addition to the white scales on the clypeus of Aedes aegypti, there's another set of characteristics that you have to check, and that is found on the side of the thorax. So these are found on the mesh epimeron. Any difference that you see? Of course, I've written it. Yes, the samples I've shown are females. All right, so the white scales, there is a question here for Aedes aegypti. There are 2 species, Aedes aegypti and Aedes aegypti. There's only, the species is the second name that you see from when you say Aedes aegypti, the species is aegypti. The genus is Aedes. But if you say Aedes aegypti, then that means you have a third name, which is the subspecies. But I am not particularly familiar with Promosis, if there is a subspecies as Aedes aegypti Promosis. So we will check on that but based on what you wrote, this is still the same species, Aedes aegypti. But there may be a subspecies Promosis, which I'm not familiar with, and we can check on that. Okay? So when you're looking when you're trying to differentiate between Aedes aegypti and Aedes albopictus, you have to look at the mesh epimeral white scale patches on the side of the thorax. So this is Aedes aegypti. And what do you notice about the best epimeral white scale patches? Are they connected or are they separate? It's separate, right? But if you look at Aedes albopictus, it is V-shaped. This is what we call V-shaped. There is no separation between the two scale patches. They are connected and they form a small V shape. However, this is not just the characteristic that you can look out for between the two species. Additional characteristics will be found on this specimen. So the one with the blue background, can you tell me what mosquito this is? This is from the side view. Can you guess? The one with the blue background? Yes, this is Aedes aegypti. Why do you say this is Aedes aegypti? What do you see? Any characteristic you can mention that makes this Aedes aegypti? Okay, possible. Yes, the mesh epimeral white scales are separate. Also, I want to mention that when you look at the abdomen between the two species, look at the abdomen. What is the difference between the two? If the first one is Aedes aegypti, so most likely I presented Aedes albopictus on the other side. So what do you see from the abdomen? There is, yes, a clear V-shaped mark on the Aedes albopictus for the mesh epimeral white scales. Yes, thank you, Minu. Abdomen for Aedes aegypti is covered with a lot of white scales. That is Aedes aegypti. So if you compare the abdomen of Aedes albopictus, there are just basal bands on the ventral side of the abdomen. So in addition, I think somebody mentioned this already, you have to also look at the legs. So can you tell me which set of legs is for Aedes aegypti? Is it the upper picture or the one lower? Yes, you're correct. Sad, there is a lyre-shaped marking on the picture of Aedes aegypti. Any comments on the legs? Which ones are for Aedes aegypti and the other, and which one is for Aedes albopictus? Very good. The upper one is Aedes aegypti. And why is this so? Can you tell me why? If this is Aedes aegypti, but you are correct. Okay, so I'll show you. For the middle leg, for Aedes aegypti, there is this narrow strip on the femur of the middle leg. So that is the second pair of legs. But for Aedes albopictus, you don't see that narrow strip of white scales. So that narrow strip of white scales tells us that this set of legs is Aedes aegypti. And then on the lower leg, you don't have the white strip of white scales for Aedes albopictus. Okay? So, although we know that Aedes albopictus will have this single longitudinal white line on the scutum, but there are many species of similar characteristic. So at least for Aedes albopictus, right, we know this is the what we look out for, this single white line on the middle of the scutum. But then to make sure that this is really Aedes albopictus, you have to check the bands on the tergites. This is the dorsal side of the abdomen. So it should be complete basal white bands. So these complete basal white bands is not always present on mosquitoes with a single longitudinal median line on the scutum. So these are some examples of mosquitoes. Maybe it's not found in your country, but these are common in the Pacific Islands and the Oriental region. So we have Aedes malayensis, Aedes henselae, and Aedes polynesiensis. All of them have different forms of the not basal, different forms of the white bands on the tergites. So for Aedes malayensis, which is also present in Singapore, along with Aedes albopictus, there will be incomplete subbasal white bands. When we say subbasal, it means it is not touching the line of the segment of the abdomen. So it is slightly away, and then since it doesn't complete one line, it is considered incomplete. On the other hand, this is also true for Aedes henselae. Good thing we don't have henselae, but it's also found in the Pacific Islands. It has incomplete basal white bands. So it must be very clear that you're looking at basal or subbasal. Basal is touching the segment, the line of the segment. Subbasal is slightly away from the line of the segment. So if you notice, all three have incomplete bands, Malayensis, henselae, and polynesiensis, but not all three is subbasal. So henselae is basal, white band, but for Malayensis and polynesiensis, it's subbasal white bands. But for these two at least, they do not come together here, but they do come together in Pacific Islands. So I would suggest that you be more familiar with the different kinds of mosquitoes present in your country, as there may be some species that may be morphologically similar and can confuse and make your identification inaccurate. Okay, thank you for the suggestions. Right. So if just as an example, right, since Malayensis and Aedes albopictus are both present in Singapore, I would like to show you some of the differences that they have that make that allow us to separate the two species. So for Aedes albopictus, the patch of white scales on the mesepimeron forms a V shape, as we mentioned earlier. But for Malayensis, it doesn't form a distinct V shape because the line is not connected or complete as that of the albopictus. But there is also these white scales on the side of the thorax, but they do not, for albopictus, they do not form a continuous line up to the scutellum. So what do I mean? So following these lines, right, these scale patches on the side of the thorax, you will notice that it's broken by spaces with no white scales. This is not because the sample is degraded, but rather because it's really a discontinuous line. But if you compare with Malayensis, the white scales extend up to the scutellum. So it forms these distinct two parallel lines. And actually, when you look at the sample from a distance, they look like it's a complete continuous line from the pronotum down to the scutellum, at least for the first line of white scales. Another thing that is very distinct between the two species is that when you look at the abdomen, this is from the side of the abdomen. So you will see that this is, I'm pointing to the line of the segment. So if the white scales touch the line of the segment, that is called basal band. So for Aedes albopictus, it is touching the side, the line of the segment. So this is what we call complete basal white bands, because if you look from the dorsal side, as I've shown earlier, they form a complete line. So that is a complete basal white band. But for Malayensis, Aedes malayensis, the white scales do not touch the line of the segment. So this is what we call subbasal white bands, but it does not form a complete line. So this is what we call incomplete subbasal white bands. Okay, so it is very confusing, especially when you're dealing with larvae. So we've found some ways to differentiate larvae, but it is very inconsistent. So it is still best to look at the adult samples before we can determine that this is definitely Malayensis and not Albopictus, or this is definitely Albopictus and not Malayensis. All right. So when we're identifying dengue vectors using different traps, there may be challenges and pitfalls that we encounter in terms of identification. So how we collect these mosquitoes is that, let's say for adult traps, we have the Gravy trap, we have the BG Sentinel trap, and the night catcher, which is actually a form of a light trap. These are good traps to have. You can also get good samples from it, but each one has its own challenge to present to us whenever we collect samples. We also can aspirate from resting areas. We can use a bazooka, but all these traps or collection methods will actually cause some of the scales to rub off or drop from friction from the sides of these collection or collection tools. So, but when we do trapping, right, it is always important to have complete locality data, because if you don't have locality data, it is very difficult to use that information. You just know that they are present, but you don't exactly know where. So it will be best if you have complete locality data, if you have a specific breeding site data, much better, so that we can have a more complete data set where we will find these mosquitoes, when do we expect them, and in what kind of habitats will they breed. So we can target that for our vector control efforts. Yes, we have shared an identification key for mosquitoes related to dengue transmission in the OneDrive. Yes, it is already posted. Thank you. All right. So when you collect different from different traps, you will collect different insects. It's not always just mosquitoes. So this is just a very small sample. Can you guess how many mosquitoes are in it? You can do direct observation of samples as soon as you uh, collect them, but definitely check first. Check first if your mosquitoes are alive. If they're alive, or if there are other insects inside that are alive, always kill them first. You have many choices of killing, but our option that we take is always to freeze them. Why freeze? Because when you freeze samples, you actually retain the taxonomic characteristics, and also they do not become brittle too fast, because some killing agents like um, chloroform, if it's still in use in your country, can cause the mosquitoes to become brittle, so then the legs will fall out. We can also use hot steam to kill them or expose them to sunlight, but always make sure that they are not able to escape your container. I think there are digital mosquito apps for identification, but the problem with some of them is that you need good internet connection. So you can go to wrbu.org. They do have good photos, and they do have good keys for identifying mosquitoes based on photos. And you can actually zoom into your region using the WRBU identification keys. Okay. So the BG Sentinel trap, for this kind of trap, you have different parts to it. So there is the lure. You can also add CO2 if you like, if you want to enhance the capability of your trap in collecting mosquitoes. So the black circular structure on top is actually the one that holds the catch bag. From here, the mosquito will fly in, and then it will be stuck by the fan below. But if you leave the catch bag in the trap for too long, then actually some samples can be too dry, and most of the legs can fall off, and most of the scales will really drop off. So this is an example of a mosquito from the BG. Can you guess what mosquito this is? Definitely, it's an Aedes mosquito. You can see from the short maxillary pulpi and the white scales on the apex or tip of the maxillary pulpi. Other than that, can you tell me what species this is? Yes, it is an Aedes female. So if you look at the clipius, do you see anything? Yes, thank you. This is Aedes albopictus because the clipius, and it is a female, the clipius is bald, it has no white scales. Then here is another sample. There are some remnants of scales, basal white scales on the tergites. So your guess is? Yeah, sorry, the photo is yellowish, but you are correct. This is actually Aedes albopictus. The yellow color of these photos is because of the light that we used at the time. The light was yellow. So this is detected by the phone camera. So samples from gravity traps, as we've mentioned earlier, will cause real damage to your mosquitoes. So these are collected using pieces of plastic straw. So actually, the good thing about the plastic straw is that it is able to scoop up your sample from the sticky lining. But at the same time, when you put it in the vials, it actually serves as a protective shield from it be sticking to the other samples that you have in the same vial. So you can actually have quite a few samples inside one vial, at least one per straw. Then you can have at least 5 straws inside these small vials. So these are some samples of mosquitoes that we get from gravity traps. So what do you notice from these samples? Are they still complete? Most have missing appendages. Most have don't have the proboscis. Most don't have legs, don't have wings, but definitely we can still identify them. That is true. Because, as I've shown you earlier, there are taxonomic characteristics that are found or at least maintained in these samples, even if they are already quite damaged because of the sticky lining. So, for the first column, what is your guess? These are 3 different species. What is your guess for the first 2 samples? All of them are Aedes at least. Okay, one mentioned Egypti. That is true. The first 2 samples are actually Aedes aegypti. If we zoom in on these photos, we can actually see the separated mesepimeral white scale patches. And if you look at the abdomen, it's almost completely white. So this is definitely Aedes aegypti. The second one, that is true. The last one is Aedes malayensis because of what made you say that this is malayensis? You can actually see the 2 parallel white lines on the thorax, and you can see incomplete bands. Yes, those parallel lines on the thorax tells us that this is malayensis, and you have the subbasal uh, incomplete bands on the dorsal side of the abdomen. But the middle one, yes, thank you. The middle one is actually Aedes albopictus. You can see that the the basal bands on the tergites are complete. And if you look closely, you can actually see the V-shaped mesepimeral pale white scales, mesepimeral white scales on the thorax. Okay, yes, elbow has complete bands on the abdomen. So we just remember all these characteristics over and over again, we can still identify damaged samples. But I do understand that there are some samples that are really damaged. And if you're really interested in identifying this, maybe you can explore molecular identification, because there is also always a limit to what morphological identification can do. Right? So that is Egypti, this is Aedes albopictus, and the last one is Aedes malayensis. All right. So for the CDC Like Trap, right? This is a very common tool. We deploy this, let's say, for 12 hours, and after you deploy this trap, you will have abundant samples, and these samples are a lot of different kinds of insects, and sometimes there's just a few mosquitoes inside. So many are non-mosquitoes, and you really have to dig through a lot of these non-mosquito samples to find your mosquito samples, because sometimes deployment takes too long, and some of the samples are really degraded by the time we receive the samples. I don't understand the question. If the pre-spiracular area has two separate spots, this is Aedes albopictus. Pre-spiracular area, not pre-spiracular area. This is actually the mesepimeral scale patches, and this is behind the mesocat episternum, which is actually the structure in between the post-spiracular area and the mesepimerum. Okay, so yeah, this, if you're pertaining to the post-spiracular area, for Aedes albopictus, it should be the mesepimeral scale patches should be connected. So it should not be two separate spots. That will be for Aedes aegypti. All right. So another sample of a trap is the night catcher. This is actually very similar to the CDC Like Trap. The primary difference is that it can be timed to rotate every hour for the next 12 hours, 24 hours. If you like, you can you can change the time intervals between collection bottles. So when you do this, you actually increase the chances of getting better mosquitoes, and also getting more mosquitoes to go inside this trap. So this is one sample of a mosquito that we collected, and we can actually use this for curation purposes, because the night catcher does not blow. The fan of the night catcher is not blowing continuously on your sample for very long periods of time. It would be between 1 to 2 hours, depending on how much time interval you want to put between collection bottles. And this allows your mosquito to actually go back to the lab still alive. And when that happens, you actually have very good. For the night catcher, at least, because of this rotation of the bottles, you change the bottles every hour. And also we add this CO2 to it. And then when we do that in combination of CO2, and then the rotation of the bottles for different time intervals, we are able to simulate uh, the human, not the human landing catch, but rather the, what do you call this? Because I have a different term for it. The use of the double net with a human bait inside. Okay, because it simulates one person standing there and doing bare leg catch. It is able to attract more mosquitoes. If we add on different lights, then we actually, if you see from this picture, we added some lights to it. We actually are able to attract other insects of importance to public health or disease transmission. We were actually able to collect a lot of biting midges and sandflies using the same trap. Yes, CO2 is an attractant for the night catcher, but we can also add lights. So just like the CDC Like Trap, the original uh, unmodified night catcher has the same light bulb as the CDC Like Trap. But the difference is that for the night catcher, it changes collection bottle at different time intervals, which protects your sample from being damaged through the whole duration of the collection period. So when we are dealing with dengue vectors, there will always be challenges and pitfalls, even for larvae. So when we do, oh no, we cannot make a mosquito wet when pinning. We can rehydrate using what do you call this? A moisture a moist chamber, but we don't make it wet because when you make your mosquito wet, then you also damage the the taxonomic characteristics that will help you identify it. So what we use is just a moist chamber to just relax the legs, relax the body of the mosquito so you can position it properly. But there is a risk to using the moist chamber. If you don't do it properly, then you actually run the risk of, because this is high humidity chamber, then you run the risk of having your samples damaged by molds. The parallel band is actually a character for many mosquitoes, not just Aedes. However, when you combine it with the characteristics that define a mosquito as Aedes, then it becomes a very specific characteristic for certain species of Aedes, such as Aedes malayensis. All right, if you're dealing with samples that are larvae from the field, you have to prepare materials before you start your identification process. Make sure that you have a clear work area, and then from this picture, we always arrange our materials before we start. So we usually have this compound light microscope. We have glass light. We have cover slips, dissecting pin, the ceramic bowl need not always be a ceramic bowl, but what we use a ceramic bowl because we need to see the sample from a white background, and the ceramic bowl what is the one that's readily available. But in your country, maybe you have enamel bowls. Anything with a white background would be very good because I'll show you later that it's important to see how they swim and how they move in the water before you even start your ID. And then always prepare, which I didn't put in this picture, always prepare a container where you can put your wastewater. Because if you're dealing with a series of larvae samples, make sure there is some place where you can throw the use the or dispose of the water from the previous sample so that you don't mix up your samples. And I always have, even if I've been doing this for quite some time, I always have an identification key with me. Sometimes you have to use the key that is common for your region, but it will be best if you can develop your own keys from your own country because that way it will be specific to species that you have in your area. You are right. Aedes is diurnal, or they are day biters. But when we are using the night catchers, we are actually able to collect some Aedes albopictus. So it doesn't mean that they are day biters. They will not. They will stop feeding as soon as the sun sets. For Aedes albopictus, I think there are some papers that will show you that they are able to feed at a certain light intensity. So you have to see that if you collect Aedes albopictus, we've done some trials where we tried to collect mosquitoes for 24 hours for a period of, I think, certain number of months. And when we did that, we realized Aedes albopictus will feed or are host seeking for at least 23 hours in a day. The reason is that when you have like available light sources, like artificial light sources, like lampposts or light bulbs, there is a certain level of luminescence that will trigger for them to still feed. However, we did not see that for Aedes aegypti. So it doesn't mean that they are day biters, they will stop feeding at all as soon as the sun sets. So you can explore this in your research. And then when you're dealing with your samples, right, going back to this, you can arrange your samples in a tray so that you don't mix up any of the samples. We are setting the night catcher before sunset because you cannot set it at night. We have to leave it there. So it should be starting to collect just before the sun sets. But there is, if you watch the video, there is a PDA where you can actually set it at what time you want it to start collecting. It will not turn on until the time that you set it. But you can deploy the trap in the site before they start collection. All right, so these are when we do samples, larvae samples, we set them up this way so that it's more organized. So the ceramic bowl is just showing you that there is a white background for you to view your larvae. Because sometimes the water in which the larvae comes in is so dark, you need this white background to visualize your sample. Okay, so there is this question. Yes, we can buy. Yes, that is true. BG is actually designed for Aedes mosquitoes as well as OV traps. But some other traps do collect Aedes even if it's not designed for Aedes. Nightcatcher is actually good and it was designed to look for Anopheles mosquitoes. So when you have your samples in a vial, pour your sample into the bowl and make sure you flush out all contents from your sample vial and make sure that no smaller larvae is left inside. This way, you can view your how your sample will swim in the water. So this is one L4 or fourth instar larvae. And then you can actually, after watching them, you can actually aspirate them one by one. Then you can put on the slide. Make sure that you're careful when you're putting on the slide so that you don't push it out too fast, that the water will spill over onto your absorbent material along with your sample. You may lose your sample that way. Okay. So when you're doing, this is what we call the temporary wet mount. So when you're doing temporary wet mounts, you're just using water. And this is how you position your cover slip. So one side is actually touching the glass slide and try to put the droplet of water in the middle of the cover slip. Hopefully your larvae is also in the middle so that they don't squeeze out from under your cover slip. And then you can use a dissecting pin to lower the cover slip slowly onto your glass slide so that you prevent bubbles from forming. Then you can view them using a binocular compound microscope. Always use a scanner first because this way you can have an overview of your whole sample or at least a good part of your sample before you zoom into the parts using the low power objective. Usually we don't examine all, but if you just have a few samples, best to examine all. But we have developed a criteria for examining samples. We always check for different colors. That's why we pour in the bowl. We check for different colors of the larvae. So darker ones, lighter ones, different sizes, some small, some big. We always collect 3 of each. And then, of course, the different stages. If you have pupa, also check the pupa. If there is pupa, check all pupa. If there is larvae, then you can check 3 samples of the different kinds that you see. So there may be a combination. We've encountered combinations of 3 different species in one sample, and there are just 3 samples inside. So it's a risk actually when you don't view everything, but of course, manpower and time constraints will actually force you to prioritize and do your identification smartly. There's a question here. At night setting, CDC light is possible to catch both Aedes since.
Mahjalia, we need
Yes.
minutes left until the end of this session.
Okay, I'm almost done. Thank you. So these are the common larvae that you'll find. So you'll have Aedes and Culex. So I'm not saying this is Aedes what species, but these are the 2 that will usually be found. You have different instars. So you will have first instar, second instar, third instar. But the best to use will be the third and the fourth instars. So when you are presented with a larvae sample, to know that they are Aedes species, you have to look at 3 things. First, you look at the siphon. Is it present? If it's present, then there's a likelihood that this is an Aedes larvae. Then check if they have pecten teeth. So pecten teeth is also on the siphon. If they have pecten teeth, then chances of this being an Aedes or a Culex is high. But to know that this is Aedes, check if you have a single pair of ventral hair tuft. So that will tell you that you are handling actually an Aedes larva. So this is a sample of an Aedes larva. So you can see that there is a siphon. Okay. There is pecten teeth. And even if it's not very visible, there is 1 pair of subventral hair tuft. So I'll just do that again for you to see the presence of the hair tuft. If it is a Culex mosquito, there will be more than 1 pair. Okay, so this is very common to look at the thorax first when you have larvae, if you want to differentiate between Aedes aegypti and Aedes albopictus. So on the thorax, because it's already viewed from the dorsal side, you look at the presence of these thoracic spines. If it is present, it's Aedes aegypti. If it's absent, then it's Aedes albopictus. Then to confirm that you're dealing with an Aedes aegypti mosquito or Aedes albopictus, then you have to differentiate and look at the comb scales. So zooming into the comb scale, this is what we call the trident comb scale of Aedes aegypti. If you use high power, if you're confident to use high power, then you will see that it has a very thick middle spine and 2 smaller lateral spines. But if you compare that to the denticulate comb scales of Aedes albopictus, when you zoom into it, it has very small lateral spines, so that when you view it, you just see 1 singular spine. And then if you have pupa, right, you can also differentiate between the 2 species. So Aedes aegypti will have this serrated or small margin of the petals and branched hairs on the last segment of the pupa. But if you're dealing with Aedes albopictus, there is a fringe of hairs surrounding the petal. You can actually use L3 for, before the break, I will just answer a few questions. You can use L3 for ID, but L2 and L1, sometimes we let them grow a bit so that we can see the characteristics better. Any study about night and day collection ratio? So it will clear that increasing night binding. We have not done a lot of checking for between night and day biting, but I have done something else in relation to malaria investigation, and it does not clearly show, because this was not one of the objectives of the study. So maybe you can look through some literature about this, maybe there is something out there. I think before going, that is true. Aedes is short and thick. However, um, sometimes looking at the size is actually very relative. So you have to look at specific structures that tell you that this is Aedes, this is Culex. I think the WRBU.org will have some electronic keys for mosquitoes, and I think I've seen some for other institutes. Yes, for some species, color, shape, and size is important, taxonomic character. But I think the more important characteristic will be the teeth and the hair tufts that you'll find on the siphon. But always remember, you just don't use one characteristic. Use a combination of characteristics to be able to differentiate this. You can actually differentiate siphon size by measuring the width of the siphon in relation to the length of the siphon. So you have to take measurements between the two because that is the ratio that will tell you this is long and this is short. Usually the shorter siphon will be the length is just less than double the width. So these are the things that you will commonly read through taxonomic descriptions of certain species. So different institutions may have different ways of how they store mosquitoes, or sometimes you don't even store the mosquito samples that you have. However, it is good to store your mosquito samples, especially when you encounter some very rare species. So Aedes albopictus, Aedes aegypti, we do have some in our collections, and we use it for teaching, but uh, this is not considered a rare species. But we've encountered some mosquitoes that we only find once in 8 years, 12 years, and just one. So for those species, we would really like to store these mosquitoes. All right. So when you are handling immature mosquito samples, how do you choose what to use? As we mentioned earlier, you can do many things. So temporary mounts, you have semi-permanent mounts and permanent mounts. For temporary mounts, these are actually using just water as a medium. So it will dissipate fast. Your samples will actually decompose, so it cannot be used for storage. But it's very useful for direct observation, and if you want your results fast, then you can just do temporary mounts. But if you want to use your samples for teaching, or let's say as a record of what you've seen, and when you're doing let's say you found a new species that you've never recorded before, then you can either do semi-permanent mounts or permanent mounts. What's the difference between the two? Semi-permanent mounts are actually using what we call hydrophilic mounting medium, or these are the ones that will actually attract a water, and when they start becoming more watery, then they start evaporating as well. So for this one, it actually requires you to put a sealant around the cover slip so that you can slow down the evaporation process. But this is what we call short to medium term storage, because for some samples, you can actually do remounting after using these semi permanent mounts. But for permanent mounts, then this one is actually can stay there for a long, long time. In my former institution, I've seen some samples from the 1930s. So imagine how old that sample is, and they're still okay. And it was done in Canada balsam. So these are what we call hydrophobic mounting medium. So they actually try to uh, move away from water, so they don't absorb any moisture from the environment. And the only thing is that for Canada balsam, it turns more yellow through time. And also, some of the components for using Canada balsam is no longer advised, like let's say use of xylene. So we've been using lately Uparal mounting medium, and I will show you later how it's done for the whole mounts of larvae and for the skins of the pupa. So this is what we use for long-term storage, and you can opt to use whatever is available in your area. So when you're trying to establish taxonomic linkages, this means that you're trying to determine the different morphological variations within a species. Let's say in your country you have not found a certain species and you found it for the first time. You want to know what are the various forms this type or species of mosquito will have, because there are some species that belong to the same species group, but they have different, let's say, formation of certain hairs, or let's say markings on the pulpy, so or the proboscis. This was seen in, and this was actually confirmed by molecular identification. Before they thought like this Anopheles punctulatus was one form, oh no, different subspecies, but turned out it's just different forms of the same species based on barcode. So when you're doing this establishment of taxonomic linkages, you start with collected larvae and you put them in individual containers. So one container, one larvae. Then since you want to have the complete set from larval stage to adult stage, then you get the larval exuvia. This is actually the skin or the molt of the larvae. You collect it. This is very hard to handle because it's very fragile, can easily break. So you usually use a scooper or a flattened piece of maybe like a flattened end of a pin, and then you can scoop it out of the water very, very carefully because this will really break up. And then you can mount this on a slide. And then after a few days that you get your pupa exuvia or the skin of the pupa, this one you will also mount so that you can know if there are many, if there are any existing morphological variation for this species. Then, of course, at the end of it, you collect your adult. You can pin the adult, and if you are uncertain of the species because it's a new record, let's say, just just an example, you can actually collect or take one of the legs of your mosquito. If you have to take a leg of your mosquito for molecular screening, always use the middle leg. So then, because the middle leg is of very little use in morphological taxonomy, so you can sacrifice the middle leg and you can use it for barcode or DNA barcoding. Yes, actually, I will show you later that you can actually put your samples in alcohol, but don't use pure or 100% ethanol because this will dehydrate your samples. However, it is true that your larvae will also dilute your alcohol because when you transfer the larvae into your ethanol, you carry over some of the water from the habitat and then you dilute your ethanol with it. So we actually use a higher concentration, higher than 70%, so that when you preserve your larvae in it, you're sure that it will not go lower, the concentration of your ethanol will not go lower than 70%. All right. So when you're doing slide mounting, you have to prepare your materials as well. Usually, slide mounting is done under the stereo zoom because you're not looking at taxonomic parts. You just want to make sure that you're putting your sample in the correct position on the slide. So you have these. You have a pipette. You have your dissecting pins. You have your fine tip forceps, and then you also prepare your cover slip and your glass slides. Always have your things ready on hand before you start your slide mounting procedures, because sometimes when you leave, let's say you leave your sample already in your mounting medium, then you have to look for, let's say, your cover slip afterwards. Then it starts to dry up and then or start moving around, then you don't have the perfect specimen that you want to present in the first place. So you can also have your mounting medium of choice. And I also included here an alcohol lamp. This alcohol lamp is actually good for removing some bubbles, but do not put your sample too long over the flame, because then it will boil over. Then, of course, it's worse in terms of the number of bubbles that you create. And then after mounting your sample, you have to view it under your compound light microscope just to see if you display the taxonomic characters to the best. Or you want the comb scales to be fully presented, or is your sample a bit skewed to the right or to the left so that it is not fully displayed in your sample? So better to use a compound light microscope to see these microscopic parts. All right. So when you have your larvae, you kill it in hot water, not in boiling water. Usually we prefer to use 50 degrees centigrade. Why is that? The reason is that when you kill with hot water, uh, that is too hot for the larvae, they tend to have this extended uh, neck from the thorax to the head. So there will be this extension of the neck, and also the sample will darken through time. But when you use just the 50 degrees centigrade hot water, then you have a nice sample that is not distended, and is a good example of your specimen. Formalin. Formalin, I haven't tried it, but I don't think it's recommended for these very small samples. Because if I remember right, 10% formalin will cause your sample to be brittle, and then you can no longer mount it. If you want to mount your sample, better to use ethanol for storage, because once you put in formalin, it will be very, very rigid and brittle. So after you kill your larvae, you can put it in a series of alcohol washes. So this will dehydrate your larvae. Don't put immediately in 100% ethanol, because that will immediately, what do you call this? Not dehydrate your larvae. Then your sample will not look very good on your slide. So use a series of alcohol washes for 80%, 90%, at least 15 minutes each in each wash. And if you think your larvae is too dark, you can actually soak it a bit for in acetic acid. Then, once your larvae is properly dehydrated, you can put it on a glass slide. So try to lift it out of the petri dish, and put it on a glass slide in a drop of euphoral essence. Don't use the pipette to transfer your larvae, because that will carry over some of your alcohol, and it's not going to look nice on your sample when you mix up your euphoral essence and alcohol. So for the once in the essence is just to make your sample. It will allow you to arrange the hairs and the structures of your sample without causing it to dry up. In doing so, it will be more malleable, as long as it's not boiling. But for us, 50 degrees work. If 60 degrees is okay for you, and it doesn't cause your sample to distend, then that is okay. We we use the 80% alcohol on the assumption that your larvae is also filled up with water. So that will dilute your alcohol. So once you have your sample arranged on the slide under the euphoral essence, because your siphon is actually pointing upwards, then that means your comb scales are not fully displayed. So what you have to do is cut between the sixth and the seventh segment. You can cut closer because your sample is actually on the eighth. The comb scales is sorry, it's already on the eighth segment. But if you don't cut carefully, cutting between the seventh and the eighth is actually too close. and you might cut your comb scale. So better to cut a little bit higher at the 6th and the 7th segment. Then you can position your siphon and lie it flat on your glass slide, and then you can display your comb scales better. Actually, yes, I've seen some literature that says that you can use glycerin in handling your specimen because it will not dry up your sample while you are trying to position the hairs and other structures on the slide. But I'm sure there is a different way in terms of how you're going to mount it when you're using glycerin after glycerin. So that one I am not particularly sure, but we can explore it. I've seen it before that they do use glycerin. Okay, and then once you positioned your sample nicely with the hairs in not that are not overlapping, you can remove the excess euphoral essence because this is a solvent. It will prevent your mounting medium to dry up if there's too much of the essence left behind. So you can remove the excess with absorbent material like tissue paper, and then you can add a drop of euphoral mounting medium. Previously, I thought it will be just 1 to 2 weeks in the oven, but it seems that it takes much longer for it to dry up. And I've read from the WRBU article that we uploaded in OneDrive, it takes them 1 to 2 months in the oven to fully dry up their specimen. So when you mount your sample on the glass slide, you also have to label it. You have to put the locality label and the determination label. Locality label includes where the sample was collected, who collected the sample. If you can put GPS coordinates, that will be also good. And then when it was collected. This information is actually important for when you're doing mapping for geographic distribution of different mosquito species. So this does not just apply for Aedes or dengue vectors. It also applies for other mosquito species. If you want to know what is available in your area and where and when, at what times of the year they're present, always put this information on your glass slides that you're keeping for future reference. And the determination label will include the species uh, that you found, what species that larvae is, and who identified it and when it was identified. So usually it is recommended that you put your larvae dorsal side up. That means when you view it under the microscope, the dorsal portion is the first part that you'll see. If you look at the head, you're looking at the dorsal part or the back of the head and not the mouth brush. If you're seeing the mouth, then you put it wrongly because you're then viewing the ventral side. And once you put the euphoral essence over your larvae, you don't have to put the cover slip immediately. You can actually let the mounting medium dry up without cover slip overnight for 24 hours. That way, it will harden a bit, and then you don't have to put these broken glass slides to make it balanced. So it will form something like a plateau on top of your larvae, and it extends the the portion that will support your cover slip. So it's not just the larvae acting as a fulcrum on top of your underneath your cover slip. The whole the partially dry mounting medium will actually help flatten your cover slip once you put it after 24 hours. So how you do it is that you put another drop of euphoral medium uh, under the cover slip before putting it on the partially dried sample that you've put there the day before. Also, remember you cut between the 6th and the 7th segment. If you let it dry out a bit overnight, uh, it prevents the siphon from moving too far away from the body of the larvae. So this way, you have a better control of your sample because once you put on the cover slip, it will push your sample to go where the mounting medium will flow. So that by allowing the mounting medium to dry a bit, then you prevent that from happening. So normally we will put the head of the larvae down and the siphon up, because this way, when you view it under the compound light microscope, you're actually going to view it right side up, because the image will invert once you put it under the microscope. You cannot preserve your larvae in 50% alcohol, because 50% will actually, in due time, they will actually decompose, because it is much, much more water than will preserve your larvae. We select the 6th and the 7th segment. It's because we don't want to cut the comb scales. The comb scales are on the 8th segment. If you cut between the 7th and the 8th segment, you actually may accidentally cut your comb scales. So to prevent that from happening, you just move one segment up and cut between the 6th and the 7th. Yeah, we will try to come up with a video for better viewing later. Once we have the bandwidth to do it. Okay, so for the any questions? Sorry.
Yes.
I think it's better if you type the question, please, in the chat box for time's sake. Thank you.
Okay, thank you. So if you're viewing the if you're viewing the skin, pupal skin, you have to separate actually the head and the abdomen, because the pupa is actually coma shaped, right? And if you don't separate the head and the abdomen will be actually sideways on your slide. So you will not be viewing the dorsal side, you will be viewing the lateral side of your sample. So by separating it, you actually view the dorsal side of the abdomen, and then you can open up the head or the cephalothorax, split it open, and make sure you're viewing the dorsal side. How do you know that? You will actually see the trumpets on top of the cephalothorax. So then you know that you're viewing the dorsal side. So once you've separated and split open your cephalothorax, then you can reconstruct the pupal skin. So once you usually do this under euporal essence. And then you remove the excess euporal essence, just like with the larvae. Egg mounting. You can actually mount the eggs the same way. It's just that eggs are harder to prepare. I mean, to find, unless you are rearing some eggs in the lab. So normally we don't prepare mounting for eggs. All right, and then once you remove the excess essence, then you can put a drop of euporal mounting medium. Again, we usually leave it exposed, in the oven for 24 hours before we put the cover slip. So again, just like the larvae, we usually put the cephalothorax down with the paddles up, so that when the image inverts in the microscope, it will be right side up. Okay, for pinpointing, these are the materials that you have. Euporal essence we usually buy from BioQuip. It is not very easy to find. So but I think there are some recipes for it. It's just that the latest we heard is that it is not that easily available. You can explore. We've tried mounting medium for blood smears for malaria. For certain countries, the brand name is Entelan, but this one also takes some time to dry up. Yes, we can not all Aedes, but at least for Aedes albopictus and Aedes aegypti, you can differentiate the pupa. But that is also the reason why we keep the pupa, because we are trying to look for morphological characteristics that can differentiate other Aedes species, or at least other mosquito species. So when you are pinpointing, these are the things that you need. You need the pins, the mounting block. The mounting block will actually make sure that everything is standard of the right height without you touching your sample when you're transferring from one tray to the next. You need your fine-tipped forceps and your insect box, and usually this is done under the stereo zone. So the best quality of samples is always from reared from larvae in the lab. So we do when we have interesting samples, we or at least the rare ones, we always keep them and pin them and put in these dry cabinets. These dry cabinets are what we call we put in the dry room. Why we call it the dry room? Because we try to maintain that the environmental condition is between with the RH between 45 to 55 degrees, percent. The reason for this is that you don't want your samples, especially the rare ones, to be destroyed due to infestation of molds because when you have too much moisture in the air, that is the highest risk for your samples. These are just naturally dried specimen. We did not put any particular desiccant to make them dry. They just dry up through time. So that means at certain points they will be attractive to ants. So it will also be good that when your drawers are actually airtight and ants cannot go into them. But we still put what do you call this mothballs to prevent. To prevent further degradation, if or at least avoid the chance of them getting degraded. And also, we always change these preservatives inside the drawers once a year to make sure that we maintain the quality of the samples that we have inside the dry room. In doing so, we actually were able to have samples that were collected from the 1970s and are still intact up until now. So you have a choice when you're doing mounting for adults. It's either you do pin mounting or you do point or card point mounting. The difference between the two is that when you do pin mounting, you actually have a 360 view of your mosquito sample. It's just that because what you do is that you stick a pin between the legs on the ventral side of the thorax. But you have to make sure that the pin does not go through the scutum because you have certain taxonomic characteristics on the scutum of your mosquito. Uh, in time, however, when you keep opening and closing your dry drawers, and in your dry cabinet, the problem with that is that it will jiggle or move your sample, and sometimes with the point that the pin goes through the thorax of your mosquito, it gets bigger, and then they become loose. And then that's the time when you start having samples dropping on the base of your insect box. So it is to do this is also very difficult. It requires a steady hand, and always you have to use freshly killed sample. So let's say we want to preserve some Aedes aegypti mosquitoes. We have to wait for them to emerge from pupa, then wait another 12 hours, at least, or 12 to 24 hours to make sure that the genitalia is in the proper position for both the males and the females, and that is the time that we can pin them. And then we collect them one by one, then put a drop of ethyl acetate on cotton wool to knock them down. So the risk here is that if you put too much ethyl acetate, it can cause the abdomen of your sample to bloat. If you don't put enough ethyl acetate, they don't die. So you have to know the narrow range where you have just enough ethyl acetate to knock them down and kill them eventually, but not enough to cause damage to your sample. So as soon as you kill your sample, then you have to pin them because at this time the body is still malleable or soft enough for you to pin them. But after that, they will start getting brittle. Then you cannot pin them anymore because once you start putting pressure on your sample, the legs will fall off, the wings will fall off, then you don't have a sample that is good enough to maintain in your collection for curation. Whereas if you have samples, let's say from traps that is good enough for pinning, then you can use your card points because they may have been dead by the time you collected them, but they are still pristine and good enough for you to see the taxonomic characteristics and display them. So, but the thing about card pointing is that we use the problem with cold plates is that sometimes they wake up. They're lethargic, but if your plate is cold but your room is not that cold, they will be not it's not enough to knock them down, enough to stop from struggling while already on the pin. So I've seen some mosquitoes that is partially alive when they were pinned, and they're of course they will move their legs and they drop off from the pin, or they drop off from the card point because they keep shifting and trying to struggle out of the connection, the point that where you pin them. So it is better to kill your sample either with ethyl acetate or freezing, and then you take them out of the freezer, let them thaw a bit before you start pinning. So when you use the glue, the pinpoint, you actually sacrifice one slide. It's not like the pinning where you have a 360 view of your sample. For the card point, one side is always adhered to the card, then that one can no longer be used for your viewing for morphological identification. But this is easier to do, and of course, if you want better samples, you will do rehydration using the humidity or moist chamber. But most of the time, this is what we use because it is faster, and it can use whatever sample that is available for you as long as they have all the necessary taxonomic characteristics for identification. So when you're doing point mounting, you can use this puncher that creates single triangular card points, and you can prepare your card points well ahead of time before you do your mounting. Then at the end of your card point, you can actually put a bit of glue, and then you can just stick it to the side or one side of your sample. When you do this, you have to make sure that your sample is complete, the legs are not in the way, and you have to make sure that when you stick your mosquito, try to use the side that is less perfect than the ones that you want to show up. But when doing so, you have to also make sure that all the legs are protected. Yes, we can use nail polish, but this nail polish actually turns yellow through time. So the glue that we use now is actually the craft glue. You can use Elmer's glue, the craft version, the craft glue of Elmer's glue, because that one will stick fast and dry fast. That way your mosquito will not fall off from your card point. If no freezer is available, we use sunlight. You can have your samples in styrofoam cups or paper cups, expose them to sunlight, and then as soon as they fall or drop to the bottom of the container, then you can start collecting them back. There are many ways to kill your mosquito, not just freezing, but of course, freezing will give you one of the better results. Nail polish remover, just be careful that because some chemicals will cause your mosquito to drop their legs. And at one point when I was doing this earlier, there was suggestions to use cigarette smoke. So I tried it and then all the legs drop off. So just see if it will cause damage to your sample, don't use it. So when you're labeling, again, just like the slide mounts, you need to put a location label and a determination label. So you can put this together or you can put it separately, but as much as you please put as much information as you can, because maybe at this time you're not using your samples or you don't need to look through your samples, but usually future researchers will actually compare your samples with what they have also collected, and it would be good that you have this information for them to compare it with. Remember that with changing climate and land use, there will also be a change in the diversity of mosquitoes that you will find in your localities. If you keep them in airtight tube, actually just a few minutes, but don't leave them with your killing agent too long, because your killing jar is not supposed to be for storage. So as soon as they die, you see that they're not moving about, then you transfer them to a container with absorbent material, like let's say a petri dish lined with tissue paper, and then cover it with another tissue paper, so that, and then with a cover slip. Make sure that you just have one layer of mosquitoes. Don't pile them up too much, because that will also damage your samples before you can actually do your pinning. Right. So when you want to transport your mosquito samples, we can do it in many ways, but I will just share with you some of the ways that we use it. We do this. So let's say you want to transport one sample at a time, but you don't have um, the time to pin them. You can actually use the 1.5 ml cryo tube or Eppendorf tube, silica gel, and this is the one that you actually have in your, let's say, medicine bottles, but you can buy them separately. And then you have your round filter paper that you cut in half. So what I would do is that I partially fill the cryo tube with silica gel, maybe just 1/4 from the bottom of the tube, and then you roll up your filter paper into a cone using your fine-tipped forceps. Actually, before I do this, sometimes I would even put my labels onto the filter paper. The information that I need for this sample can be put on the filter paper with pencil, because I don't want to put additional um, tags or papers inside the tube that can cause damage to my sample. So the less moving parts that you have inside, the better. So make sure the silica gel is outside your filter paper, not inside the filter paper. And then this is how it will look like. You can see the little pieces of silica gel outside the filter paper, and we will use this to put one sample inside. So you can put more actually, but if you want to use this sample for, let's say, pinning later, or you just want to send this separately, you can. You want to make sure that it is intact by the time it gets to the place you have to present it to. So what we do is, for each tube, we actually put in a cryo box. So in one cryo box, you have, I think, 81 samples you can put inside. But if you're not maintaining the integrity or the completeness of your sample, and you're actually going to crush it for, let's say, DNA barcoding, you can put more inside each tube, maybe up to 5 inside. It will still be okay. But when you take it out, use fine forceps and make sure that your filter paper does not flick away your samples. So you can also transport adults as individual pinned samples. So this is a single pinned samples. I cannot put too much information inside, but I can still put a code number. So with a code number, I can refer to, let's say, an Excel file, and I can give information about this sample based on that number. Or I can send multiple samples. These are actually reused uh, filtered, what's this? Tips, for pipette tips, filtered pipette tips, and we reuse them for temporary storage of pin samples. And you can actually bring your samples using this method. Okay. And when you ship them, make sure that you label your sample as dead insects for scientific study of no commercial value. It is not difficult to send out dead adult mosquitoes. It is more difficult to send out larvae in alcohol. So I will share that with you in a while. Okay, so these are slide mounts, and for slide mounts you can use a slide box. But before you send out your slide box, always put this absorbent material on top to prevent things from shaking when they are being transported. And then when you have alcohol, actually ethanol is considered dangerous goods, and it's labeled as UN number 1170. However, in accepted quantities, this means in minimum quantities, it can still be uh, transported, but the inner container will only have a maximum of 30 ml, and together they should have only a maximum quantity of 500 ml. But it will take a while for you to find a good courier. So make sure you follow the shipping protocols in your country. Yes, sorry. Any question? The filter paper actually absorbs the moisture from your sample, so it prevents it from getting moldy. And then the addition of the silica gel will also dry up the environment within the cryo tube. So when you're shipping eggs, we may think that we can just ship them in a hardy container like this, but actually no. When you ship eggs, you have to put them in different containers. So here, this is a secondary container. This is a container where you have a moist cotton to maintain humidity inside the secondary container. And this is where you put your eggs. This is what we call the primary container for your eggs. So both the egg container and the moist cotton will have netting on top so that the moist cotton will not come into contact with the eggs. So you can put them together inside the secondary container. And this secondary container will actually be put in a third container, and then you can put this absorbent material around it. This way, it will protect your sample from being crushed. And actually, after putting in this secondary container, you can even put in an overpack with what we call styrofoam peanuts. This will further absorb any impact or trauma to your package and then completely protect your sample from being destroyed. But when you ship eggs, always make sure you comply with all government and shipping regulations, because we have had instances where um, we lost some samples because some of the permits were not in order. So make sure you have that in order before you start shipping eggs. And then you can also transport live larvae. So make sure that you can you have this container only half filled with water. When you do that, uh, you actually allow them to survive longer, because if you fill it up with water, you actually suffocate your larvae. I have a question here. Actually, it is better to transport as in the form of eggs, but always comply with regulations for the institute who will receive the eggs, because your samples may be destroyed in the process. Yeah, it's only for transport. The filter paper is not for long-term storage. It's just for transport, just to make sure your sample is not destroyed from when you send it out to the point where they will receive your sample. And aside from sample vials, which is what we use here, you can also use the Whirl Pack or plastic container. But again, just put water up to about half. So for the Whirl Pack, they actually put a line, white line there, up to where you can just put your water samples. Yeah. So normally we don't transport adults, but maybe for short-term transport, like from collection site to your lab, you may need to transport adults. Usually we do that by putting the adults in cold box, like a styrofoam box with some cold dogs inside or cold packs inside. This way, they don't die from heat overheating because the styrofoam box will really contain the heat. And then also, if your country is very warm, it will be good to have that protection. But the thing about adults is that they're very sensitive and it's not easy to maintain them. eh, Usually when we receive samples, when we transport adult samples, it's just for short distances. We try to make sure that it's not too far and we will not be wasting our effort. Yeah. So when we do adult collections, we try our best that we can bring it to the lab at the shortest possible time. We are trying to do that for colonization procedures, and it is really difficult. Either you collect and bring back to the lab immediately, or you do additional things like you put in sugar, in cotton wool, so that they have food in transport and those kinds of things. So for me, if I'm going to transport, I'm going to transport the immatures like the eggs and the larvae rather than transport the adult.